Hello, good morning students. So we are studying the chapter means the chapter which is in progress, that is strategies for enhancement in food production. So in this, in live class we are discussed about the plant breeding for the disease resistance. We are already discussed. But now, once again, I am going to tell you because I don't know how much means how far so far it is convinced you people. So today, once again, I am going to discuss about the plant breeding for the disease resistance. In the sense, we all studied about the plant breeding. The plant breeding technique, it is the one of the important technique that help to us in the sense plant breeders to increase the productivity, to increase the food productivity, to fulfill the need of wood for the increasing population. So now, in that means for a particular plant breeding technique, what we are going to go for, or what the plant breeders are going to go, or what they are going to do for developing the disease resistance plant. Disease resistance plant in the sense, in this particular point, plant breeding for the disease resistance. Particularly, we are going to go for manipulating the plants. See, this is important, very important. In this particular concept or plant breeding for the disease resistance in the sense, in this breeding, purposefully we are considering or we are concentrating, or breeders, plant breeders are concentrating only the character, in the sense disease, only in the sense importance. We are going to emphasize in the sense emphasis. You are going to give the emphasis, or you are going to give the importance to the disease resistance genes. Disease resistance gene in the sense, when we are going to go for the plant breeding, that time we are going to look at the plants, or when we are going to selecting the variabilities, that time we are going to concentrating on the disease resistance plant, so that we can reduce the what we are going to call as a crop species losses, in the sense of production of food loss. Understood? Now, here in this particular point, plant breeding for the disease resistance ತಂದಾಗ ಈಗ ನಾವು ಪ್ಲಾಂಟ್ ಬ್ರೀಡಿಂಗ್ ಅಂತಂದಾಗ ನಾವು ಬಹಳಷ್ಟು ಪ್ಲಾಂಟ್ ನ ಮೊದಲು ಕಲೆಕ್ಷನ್ ಮಾಡ್ತೀವಿ, ಅದಕ್ಕೆ ನಾವು ಜರ್ಮ್ ಪ್ಲಾಸ್ಮಾ ಅಂತ ಕರಿತೀವಿ. ಆನಂತರ ನಾವು ಕ್ರಾಸಿಂಗ್ ಮಾಡ್ತೀವಿ, ಹೈಬ್ರಿಡೇಷನ್ ಮಾಡ್ತೀವಿ, ಆನಂತರ ಸೆಲೆಕ್ಷನ್ ಆಫ್ ದ ಹೈಬ್ರಿಡೇಷನ್ ಅಂಡ್ ಟೆಸ್ಟಿಂಗ್ ಅಂಡ್ ರಿಲೀಸ್ ಅಂತ ಎಲ್ಲ ನಾವು ಕಲ್ತಿದ್ದೀವಿ. ಸೊ ಅದನ್ನ ಯಾವಾಗ ಮಾಡ್ತೀವಿ ಅಂತಂದಾಗ we used to improve the food production. ಫುಡ್ ಪ್ರೊಡಕ್ಷನ್ ಇಂಪ್ರೂವ್ ಮಾಡೋ ಮುಂದೆ ಅದ್ರ ಮತ್ತೊಂದು ಇಂಪಾರ್ಟೆಂಟ್ ಪಾಯಿಂಟ್ ಯಾವ್ದಪ್ಪ ಅಂದ್ರೆ ಡಿಸೀಸ್ ರೆಸಿಸ್ಟೆನ್ಸ್. ನೀವು ಪ್ಲಾಂಟ್ ಸೆಲೆಕ್ಟ್ ಯಾವ ಮಾಡಿರ್ತಿರ್ತಂದಾಗ ಅವ್ರಿಗೆ ಯಾವುದೇ ರೀತಿ ಏನಪ್ಪ ಅಂದ್ರೆ ಪ್ಯಾಥೋಜನ್ ಅಟ್ಯಾಕ್ ಆಗಂಗಿಲ್ಲ, ಅಥವಾ ಪ್ಯಾಥೋಜನ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಇರ್ತಾವೆ ಅಂತ ಪ್ಲಾಂಟ್ಸ್ ನೀವು ಬ್ರೀಡಿಂಗ್ ಮಾಡೋ ಮುಂದೆ ಸೆಲೆಕ್ಟ್ ಮಾಡಿರ್ತಂದಾಗ ಆಟೋಮೆಟಿಕ್ಲಿ ಯು ಆರ್ ಗೋಯಿಂಗ್ ಟು ಗೋ ಫಾರ್ ದ ಇನ್ಕ್ರೀಸಿಂಗ್ ದಿ ಫುಡ್ ಪ್ರೊಡಕ್ಟಿವಿಟಿ. ಸೊ ದಟ್ ಪಾಯಿಂಟ್ ವಿ ಆರ್ ಗೋಯಿಂಗ್ ಟು ಡಿಸ್ಕಸ್ ಇನ್ ದಿಸ್ ಪರ್ಟಿಕ್ಯುಲರ್ ಪಾಯಿಂಟ್, ಪ್ಲಾಂಟ್ ಬ್ರೀಡಿಂಗ್ ಫಾರ್ ದ ಡಿಸೀಸ್ ರೆಸಿಸ್ಟೆನ್ಸ್. Here, the wide range of fungal, bacterial and viral pathogens are there. Wide range of fungal, bacterial and viral pathogens affect the cultivated crop species. Cultivated crop species ಅಂದ್ರೆ ಏನು? For example, ನೀವು ತಗೋಬಹುದು groundnut, sugarcane, wheat, rice, jawar, maize, okay, then bajra, etc. Like that, those cultivated crops ಏನಾಗಿರ್ತಂದಾಗ ಇಂಥ ಫಂಗಲ್, ಬ್ಯಾಕ್ಟೀರಿಯಲ್, ವೈರಲ್ ಪ್ಯಾಥೋಜನ್ಸ್ ಏನಪ್ಪಾ ಅಂತಂದಾಗ ಅಟ್ಯಾಕ್ ಮಾಡೋ ಚಾನ್ಸಸ್ ಇರ್ತೈತಿ. and that too in a tropical climate. I told you about the tropical climate in a sense. Here we are going to get only two seasons, one is a dry season, another one is a wet season, and an average temperature in a tropical climate is 18 degrees centigrade, okay, in a month. Understood? That is what, and the tropical climate or tropical regions are the considering as North America, Asia, Asian countries, or all of these are considered as a tropical climate. Here what is going to happen due to the fungal, bacterial and viral pathogens which are cause the diseases in a cultivated crop, that can leads to 20 to 30% loss of crop productivity. What they can means they are what we are going to call as a interrupted, or they can lead to reduce 20 to 30% loss of cross productivity. In such cases, what we are going to go for? We are going to go for the plant breeding in such a way that whatever we are going to get after the plant breeding, the new variety of crop that is resistance to the fungal infection, that is resistance to the bacterial infection, that is resistance to the viral infection, so that we are going to reduce the loss of crop productivity and increase the crop productivity. Understood? That's what I written here. So plant breeders have to develop, plant breeders in the sense who all are working with the breeding, plant breeding and all scientists, those have to develop disease resistance crop to increase the food production. In the sense, if we are going to increase, means we are going to develop the disease resistance crop that can increase the food production. Only it is not helpful for the what we are going to call that as a food production, but rather than this plant breeding from the disease resistance technique, it is helpful to what we are going to call that one as a reduces the development, reduces the dependence of fungicides and bactericides. Now fungicides ಅಂತಂದಾಗ chemicals use ಮಾಡ್ತೀವಿ to kill the fungi, bactericides to kill the bacteria. Okay, ಅವತ್ರದ್ದು ಕೂಡ ಯೂಸ್ ಮಾಡಕ್ಕೆ ಏನಾಗ್ತಿಪ್ಪ ಅಂತಂದಾಗ farmers used to pay the money or spend the money. ಆ ರೊಕ್ಕ ಹಾಕಕತ್ತೈತಿ, ಆ ರೊಕ್ಕ ಲಾಸ್ ಆಗೋದು ನಿಲ್ತೈತಿ. ಆ ನಂತರ ಮತ್ತೊಂದು ಇಂಪಾರ್ಟೆಂಟ್ ಪಾಯಿಂಟ್ ಏನಪ್ಪ ಅಂತಂದಾಗ, if we are going to reduce the dependence of fungicides and bactericides or fungicides use ಮಾಡೋದು ನಾವು ಕಡಿಮೆ ಮಾಡಬೇಕಂದ್ರೆ we used to contribute the environmental condition, in the sense environmental issues ಏನಿರ್ತಾವ, pollution ಏನಿರ್ತೈತಿ, ಆ pollution ಕೂಡ ಕಂಟ್ರೋಲ್ ಆಗಬಹುದು. ಇದರಿಂದ ಪಾತ್ರನಾಗ plant breeding for the disease resistance ಏನಪ್ಪ ಅಂದ್ರೆ plant breeding for the disease resistance technique ನೇ ನಾವು ಮಾಡಿದಾಗ we used to reduce the dependence of fungicide as well as bactericide. This can it can help to improve the economy of farmers as well as it can improve the what we are going to call that as a environmental conditions, in the sense it can reduce the pollution. Right? Then next, how we are going to go for the plant breeding for the disease resistance, in the sense already there are some what plants which are naturally having the disease resistance capacity, and now if you are going to go for the plant breeding for the disease resistance, that time the breeders must and should know about the what we are going to call that as the organism which is going to cause the disease, in the sense disease causing organism, and mode of transmission, how it is going to transfer from one to the another, in the sense mode of transmission. That once you are going to know, after that you are going to go for the what we are going to call that as a plant breeding. Now here some diseases they are mentioned here which are you are going to observe in a plants, like fungal diseases that we are going to call as a rust, brown rust in a wheat, red rot for the sugarcane, late blight for the potato. These are the some diseases which are caused by the fungi. Then bacterial, in the sense black rot for the cruciferous in the sense what ನೀವು ಕುಂಬಳಕಾಯಿ ಆಮೇಲೆ ಸೌತೆಕಾಯಿ ತಿಂತಿಲ್ಲ cruciferous. Then viruses like tobacco mosaic and Trump mosaic, whatever in your area here. So many farmers they used to grow the what we are going to call as a tobacco. A tobacco may be viral disease ಆಗ್ತೈತೆ. viral disease ಗೆ ಅಂತ ಪಾಠದಾಗ tobacco tobacco mosaic MASAIC mosaic or a Trump mosaic viral disease ಆಗ್ತೈತಿ. So these are some common diseases we are going to found in a what we are going to call as a cultivated crop species. So this means for this, if we are going to reduce what we are going to call as a dependence of the fungicides, and we have to increase the disease resistance to such fungal, bacterial and viral diseases, we are going to go for the plant breeding for a disease resistance, and that is it is important in improving the productivity, agricultural productivity, in the sense what cultivated crop species productivity. Right? This is about what plant breeding for the disease resistance. Now the important point we are going to study, what is that, in the sense what are the different methods we used to follow, to successful plant breeding for the disease resistance. Here I written methods of breeding for the disease resistance. What are the different what are the methods we are going to use? One is conventional breeding technique or mutation breeding technique. Conventional breeding technique or mutation breeding technique. In these two, we are going to start. Let us know about the conventional breeding technique. Here, conventional breeding in the sense what? Conventional breeding is nothing but hybridization and selection. I think or I hope you all are familiar about the steps which are involved in a plant breeding technique. Plant breeding technique steps our steps used to follow here in a conventional breeding technique. In the sense, conventional breeding technique is nothing but hybridization and selection. Hybridization and selection. In hybridization and selection, for plant breeding for the disease resistance, what are the steps involved in that? The similar steps what you are studying in a plant breeding, common plant breeding to increase the productivity. Like that, here you are going to go for few steps which you have to follow that can help you for the what we are going to call it as a getting disease resistance plant. Getting disease resistance plant. Now here the steps involved in a conventional breeding technique. That is the first step. You all know about this. What is that? Screening of germ plasma for the disease resistance. Screening of germ plasma for the disease resistance. Already I mentioned here, what I have mentioned here in the friends, there are some plants which have a naturally disease resistance capacity. It already been naturally in some plants they are having the disease resistance capacity. So in plant breeding for the disease resistance, by the conventional breeding method, first step screening of germ plasma for the disease resistance. ಅಂದ್ರೆ ಇದರ ಅರ್ಥ ಏನಂತಂದಾಗ ಯಾವ ನೀವು ಪ್ಲಾಂಟ್ ಬ್ರೀಡಿಂಗ್ ಮಾಡೋದ್ನಾಗ ಅಥವಾ ಪ್ಲಾಂಟ್ ಬ್ರೀಡಿಂಗ್ ಫಾರ್ ಡಿಸೀಸ್ ರೆಸಿಸ್ಟೆನ್ಸ್ ಮಾಡೋ ಮುಂದೆ ಏನು ಒಂದು ಜರ್ಮ್ ಪ್ಲಾಸಮ್ ಕಲೆಕ್ಷನ್ ಮಾಡಿರ್ತೀರಿ, ವೆರೈಟಿ ಆಫ್ ಸ್ಪೀಸೀಸ್ ಕಲೆಕ್ಟ್ ಮಾಡಿರ್ತೀರಿ. ಫ್ರಾಮ್ ದೆಮ್ ಯು ಆರ್ ಗೋಯಿಂಗ್ ಟು ಸ್ಕ್ರೀನ್ ಆರ್ ಯು ಆರ್ ಗೋಯಿಂಗ್ ಟು ಸೆಲೆಕ್ಟ್ ಅಬ್ಸರ್ವ್ ದಿ plants which are having what disease resistance capacity. Disease resistance capacity. Such plants you are going to select as a parent. Such plant you are going to select as a parent. Understood? So next point is hybridization and selection of a parents. Hybridization and selection of a parents. Then you go for the hybridization. Then again you are going to go for the selection of a parents. Now next point is selection and hybridization of. Okay, right. One niche. I written 2 times. I think so. Hybridization and selection of the parents. After this, hybridization and selection of the parents, then we are going to go for selection, evaluation of hybridization and release to the new variety. In the sense, first we are going to go for screening of the germplasm for the disease-free resistance, then disease-free resistance, then after that you are going to go for the hybridization of a selected parents, hybridization of selected parents, not selection, selected parents. After that, selection, evaluation of a hybrids, whatever you are going to get the new offsprings, those you are going to go for selection, evaluation of the hybrids and test hybrids, testing and release of new varieties. After that, we are going to go for the testing and release of a new varieties. Understood? Testing and release of new varieties. So these things you know about these things. You are studied already in a plant breeding technique. In the sense, in a conventional breeding technique, you are going to go for hybridization and selection. This is the what the basic thing involved in a conventional breeding technique. Conventional breeding technique for the disease breeding for the disease breeding plant breeding for the disease resistance plant. In that, first we are going to go for screening of germplasma for the disease resistance. After that, we are going to go for the hybridization and hybridization of selected parents. Hybridization of selected parent. Please make it correct. Hybridization of selected parent. Then you are going to go for crossing or hybridization between the selected parent after the screening of germplasm. Then at last, whatever you are going to get the new offsprings, what you are desire means what you want, this is resistance plant in that you are going to go for selection, evaluation of the hybrids and testing, release of the new varieties. Then you go for the release, testing and evaluation of the new varieties. These are the what steps involved in a conventional breeding technique. Okay, next we are going to discuss about what mutation breeding technique. In mutation breeding technique, what is the mutation? Usually, I think when we are discussed with online that time, few students answered me mutation in the sense there is a sudden change take place in a genetic makeup. Sudden change take place in a genetic makeup, and how that mutation breeding is help to getting the disease resistance crop. We will study here. Okay. Next thing is after the conventional breeding technique, we used to study the mutation breeding, and already I mentioned what is meant by mutation in the sense the sudden change takes place in a genetic makeup. That is where we call as a what? Mutation. Here in a mutation breeding, what we used to do in the sense mutation breeding is nothing but the process by which the genetic variations are created. Genetic variations are created in the sense using the mutation. Mutation is sudden change take place in a genetic makeup. Using that process, we are going to go for creating the new varieties. Through the change in the basic sequence within the gene. Within the gene, we are going to change the basic sequence to get the desired gene which we want. That is what we are going to call as a mutation breeding. For example, if suppose there is one gene that is not resistance to the what we are going to call that one as a disease, that gene we don't want, and another one gene is there that is expressing or that gene is responsible for the what disease resistance. That time we are going to go for silencing that first gene, and we are going to induce or we are going to stimulate the second gene for the next generation. That can we are going to get the disease resistance plant. So that is what the mutation breeding is nothing but the process by which the genetic variations are created through the change in the base sequence of the gene. Base sequence within the gene. A particular gene we are having the sequences. We are going to change in that sequence to get the desirable gene. Desirable gene in a sense what we want. And how we are going to go for doing this, what we are going to call it mutations, are happening naturally within the organisms. Mutations are happening naturally within the organisms, and mutations are helpful for the organisms. Also. All the mutations are not harmful in the sense evolution. Whatever the evolution we are going to means study nowadays, or whatever the evolution is happened, in that the mutation plays an important role. Right now here, that is a natural mutation, and here in plant breeding, what we are going to go for, how we are going to go for the mutation breeding in the sense, we are going to introduce the artificial mutations. Artificially we are going to introducing the mutations through the use of some chemicals, and radiations like gamma radiations. Using the chemicals and radiations like gamma radiations, we are going to induce the artificial change in a base sequence within the gene to get the desirable character of what we are going to call it as a new plants, and this process is called as mutation breeding. So that this is about what we are studied in a today's class, plant breeding for the disease resistance. What are the basic things you used to know regarding the plant breeding for the disease resistance, and after that methods of breeding for the disease resistance. One is conventional breeding technique. In that we are going to go for the screening of germplasma collection, then hybridization of the selected parents, then after that whatever you are going to get the offsprings, evaluation, observation, selection and testing and releasing for the commercialization. Those we are going to use in a conventional breeding technique. Next one is a mutation breeding, in the sense what is mutation, how we are going to induce the mutation into the particular plant artificially by using some chemicals and radiations like gamma radiations to get the desirable plant, and this process is called as a mutation breeding. So these are the means few important points regarding the concept of plant breeding for the disease resistance. And now US means the homework for you people. Yesterday also in online class I told you, you used to go through the table 9.1. There they are given crop and variety of crop and disease means resistance to the disease. Whatever the crop varieties which are developed for the disease resistance, the table is given in your book, page number I think 174. That table you used to remember. That table used to remember for the examples of the plants which are developed using the plant breeding technique for the disease resistance. Okay. With this all the points regarding the plant breeding for the disease resistance, I am going to stop here. And in the next class, we used to study about the plant breeding for developing resistance to the insect pest. Here we are studying the plants which are developed for the resistance to the diseases like fungal, bacterial, and viral diseases. In next concept, we are going to study the plant breeding for developing the resistance to the pests, in the sense insect pest and other things. Okay, that we are going to discuss in the next class. So I am going to stop here with this what fruitful information regarding the plant breeding on the disease resistance. Thank you.